Okay, guys, so now is the time to move on to the actual topic. Um, so we reached here, and I told you that by the end of this topic, you should be able to understand DNA replication and everything that's happening here. So, essentially, just this diagram, making sure that you understand what this diagram is showing will be enough to understand everything that's happening. So, first things first, you should generally know what the DNA looks like. By now, you do. This spiraling is the DNA. Here, there's some work going on, which is why this spiraling is opened. As you can see here, I told you that there are two anti-parallel strands and they have a backbone. This red ribbon is the, let's start just here from this side of the diagram, okay? So there's a backbone, this red color, which was the, if you remember, phosphate and the ribose sugar. The second thing you remember is that the base pairing takes place between the bases, which are in the center, and then the DNA is obviously twisting upon itself and further twisting to form the folds and the chromatids and the chromosomes that we talked about. Now, I also told you that the difference between non-coding and coding DNAs or genes was basically coding ones are open, which means things can happen between them. Things something's happening for them, right? But non-coding ones are folded further and further to form those chromosomes. Now look at this. This is the exact prime option where you can see, appreciate how this one is coiled. However, over here, there is something that works to open this DNA up. First, it was straightened, then it was opened. So appreciate here how this DNA is opened. And then over here, you saw that there was only one strand and two strands. However, by the end of the diagram, you can appreciate four backbones. I told you backbones were the nucleic acids, and the other is just, and DNA is basically two nucleic acids bonded together by hydrogen bonding of base pairs, which is the base complementary base pairing, right? If you can appreciate, there are two nucleic acids here. However, there are four nucleic acid here. You can count that by the red backbones, 1, 2, 3, and 4. So there are four over here, which means that DNA has doubled itself. It has replicated. So the process, whatever process happened from this end to this end led to two different, not two different, but two DNAs forming, right? And this whole process going like just like the cell, where one cell dividing into two was cell replication or cell division, this is DNA replication. This is a process by which from these two nucleic acid strands we achieve four nucleic acid strands, or from this one DNA molecule we achieve two DNA molecules, right? And so the process is fairly simple. All that happens is now going to be displayed here. So all that happens is that you have a DNA, which is two chains with complementary base pairing. These chains are opened up and their base pairs are separated. And these keep on opening. The direction of opening will move towards this side. As you can see, its direction of opening is increasing. So what happens is, against each chain, free nucleic acids come in and bind. And you know that these free nucleic acids, their base pairing occurs. For instance, if this was adenine, then a thymine will come in, form complementary base pairing, and the nucleotide will attach here. And if the next one, for example, is guanine, it will form nucleic base pairing with cytosine. Now, these two molecules, these two nucleotides that have formed successful base pairing, what will happen to join them into this bond? We know that nucleic acids have a 3,5 phosphodiester linkage bond, which means, remember that those two nucleotides from here that were forming this bond. So if this base pairing has successfully occurred, now the next thing needed is for this 5 to bind to this 3, which is responsible by an enzyme, because there are no hydrogen bonds here. There are covalent 3,5 phosphodiester bonds. Hydrogen bonds are spontaneous, just like I told you. If it's like a magnet, cytosine sees guanine, the hydrogen bonds attract and fish combine together, we get base pairing. But what of these sugars? The sugar backbone has to form by covalent linkage. So that means now we need an enzyme that works, for instance, an enzyme that works to join. Look at this nucleotide coming in. This nucleotides base pair will make sure that it's magnetically attracted here and joins here. Now we need something to connect this 5' end to this 3' end, right? Or this 3' end to this 5' end. So how do I do that? I need an enzyme, something like this, an enzyme working to staple it together. So let's see what those enzymes are. Let's see everything. But first, what we need to see is that how DNA replication is taking place. So we have these two strands that are opening up, and nucleotides are coming in, bonding with each of the strands separately until we reach the end of the DNA. And when the end opens up and the last nucleotide binds, then what ends up happening is that we have two totally different strands that have no connection to each other anymore. However, because for instance, if this was one parallel strand and the other blue one was another parallel strand. Now, one thing should be pretty clear, that if this is 3' to 5', if this one, okay, let me just write it here so it becomes easier. So if this one is 3' to 5', then this blue one must be 5' to 3', running opposite. So if this one is exactly opposite to this one, and the new strand, this golden strand that forms right here, should be exactly same to this blue one, right? Only then can it form its base pairing over here, when it's exactly same to this one. And or I can say is that even this new strand must be 5' to 3', right? With the same nucleotides, because the original structure or the original sequence remains the same. So if this one is 5' to 3', the new golden one, it resembles the old blue one, which was also 5' to 3'. So what it essentially means is that both the DNAs are exactly similar. Remember that chromosome that we had? It had one strand of DNA here and one strand here. And I told you that semi-conservative replication takes place. What does semi-conservative means? It means that both of these strands, half of these strands are preserved from the original one, but the structure is overall conserved. New strands are exactly opposite to these or similar to their brothers that were separated from them. So a semi-conservative, and I'll explain this later as well, but just remember that, imagine this as the chromosome. This goes over here in this arm, and this goes in this arm, of course. And then we know that both of them are exactly the similar, which is why even when the chromosomes were cut in the center, a whole DNA gene that is identical to this whole DNA gene was going to both cells in mitosis, if you remember. Okay, so this is exactly how DNA replication takes place. The whole process, and every part, obviously, you know that whenever processes are occurring, to speed them up, we need enzymes. So obviously, to open the DNA up, we need enzymes. To make sure that once we reach here, these strands remain closed and this strand remains open, we need another enzyme. Then we need enzymes to make sure that these bonds are joining. Like I said, the phosphodiester linkages are joining, and the process is stable. No part should, and because imagine if these parts are open, slightly open, and work is going on. So if in this case there's not a lot to mess up, but for instance, if these are open, a large space is open, and the enzymes are working over here, then we need some enzymes to stabilize this portion. Otherwise, this portion will coil upon each other, and these four can form these they can literally form these weird coils upon each other and destroy themselves, destroy their original shape and structure. So we need enzymes at every point of the cycle, just to make sure that the structures are stable even by the end, until we get the final products that can then stabilize themselves. So look at this one. It is semi-conservative replication because this one is exactly halfly conserved in both it's semi-conserved, half conserved in both the end particles. Okay, let's see further what I mean by this. So again, just an image showing you the base pairing. Look at this coming in. So most probably one strand is opened from here, the other strand is opened from here, it's opposite strand. And then look at the at GC, again, we need a T. With a G, we need a C. So if I give you this strand only, and I say, okay, the base pairing of this strand is C, T, G, A. and I asked you what nucleotides do you need to put. If I give you a solution in the lab, I will ask you what nucleotides do I need to put in this, or what sequence will it form when I am forming base pair for these. So first of all, you know that okay, these represent the three or the two hydrogen bondings. For C, I need a G. For T, I need an A. Again for C, T, and again A. So if I tell you that my original sequence was a CTGAT, and I ask you what sequence will its complementary base pair have, so what sequence will it have, and I tell you this is from 3 prime to 5 prime, and I ask you, okay, what sequence does this have? So you're going to tell me GACTA, right? And then we know the primes are always reversed. So 5 prime to 3 prime is the correct sequence that we have. So I hope that is clear. Now, what ends up happening in a question is, in a question, he asks you, please write 3 prime to 5 prime sequence of the nucleotides that will form against this strand. Now you will end up writing this, and then it's going to be marked incorrect. Why? Because he's saying write 3 prime to 5 prime base pairing, which means you were supposed to start here. This is the 3 prime end. So always make sure when you are making, so when the question comes up, if he says ATC and 3 prime to 5 prime, and he says, okay, I need 3 prime to 5 prime base pair sequence for ATC. And then first what you do is you write their base pair sequences. Okay, now that I've written those, then you write the number. So 5 prime to 3 prime, obviously this anti-parallel strands, if this is running here, then it should be exactly the opposite. And then I go towards the answer and say, okay, he wants 3 prime to 5 prime. So I'm going to write GAT in the answer, which is 3 prime to 5 prime, not TAG, which is wrong. TAG is not 3 prime to 5 prime. Okay. And why is that important? You'll get to know soon why it's important. But one thing to know over here is that these three, so three of these, they work as a packet. Three of these code for one amino acid. Okay, so this is one information you need to remember. Three of these, which means each, so these act as codes. For example, they're like passwords. Your ATM pins have passwords, right? But those passwords are 4-digit letters that will tell you, okay, so my ATM pin has a password 1234. So my name is associated, Hafsa means 1234. So every time I enter 1234 with my card, it means that Hafsa amino acid has to be called out, right? In this case, the amino acids, one amino acid, so AA is amino acid short form for me. So CTG, for example, I say is one amino acid. For example, I say it stands for proline. Proline is an amino acid. So every time there is a CTG attached, it's going to, which is 3 prime to 5 prime CTG attached, it's going to attach for proline, right? So if I write this, for instance, if he has asked me to write from 3 prime to 5 prime and decide the amino acid, and I don't know the difference between 3 prime to 5 prime, and for instance, I write GTC, then is this code and this code similar? No, they're not, which means my amino acid is messed up because I'm not writing it correctly, because the correct direction from 3 prime to 5 prime is CTG. The correct direction from 5 prime to 3 prime is GTC, and that probably stands for another amino acid, right? Some other amino acid. So you have to make sure that you're writing the directions absolutely correctly according to the question he's asking. If he's not specifying which direction, then you can just, for instance, if he says my code is AGT, what is going to be the nucleotide code of the opposite strand, or the parallel anti-parallel strand, then you can just say TCA. Opposite to the AGT is TCA. Opposite to AGT is not ACT. It's always TCA because of the anti-parallel strands. Just make sure you read the question well. And again, as these come in, you need an enzyme to form a covalent 3,5 phosphodiester covalent bond here, right? So that is one thing in DNA replication. I hope you guys have seen the video because it's a very good illustration explaining these. And so then I told you I need an enzyme to form this backbone with this backbone. And what is that enzyme? So for instance, let's just stick with my pointer here. So my pointer says that I base paired these, I base paired these, I base paired these, the backbones were formed. But then an incorrect, a CT, we know A and T always form. So an CT base pair becomes, now the enzyme that was supposed to form this bond over here, this 3,5 phosphodiesterase bond that was forming this bond, what should it do? Should it form this bond or should it not? Let's see the property of the enzyme. First and foremost, that enzyme is called DNA polymerase. This enzyme, this green and yellow, is the DNA polymerase. The green site is its active site, okay, just to show you where the bonds are forming. And the word DNA polymerase speaks for itself. DNA is this molecule, and polymerase means that a polymer is being formed. And whenever we have an enzyme, it ends in an ase. That I hope you remember that concept from O level. So DNA polymerase is a specific enzyme for this. Now, what does DNA polymerase do? DNA polymerase forms this 3,5 phosphodiester linkage, right? And this phosphodiester linkage is, which means that not only is it synthesizing this linkage, but it's also responsible for making sure that if a mismatch comes in, it does not form this linkage. Instead, it throws it out and waits for the right nucleotide to come in. So what are the two functions that DNA polymerase does? The first function is that it's going to synthesize these 3,5 phosphodiesterase bonds. And the second function is that it's going to proofread. It's going to make sure if a wrong nucleotide comes in, it does not form the 3,5 linkage. Instead, it spits it out, or it excises it. It takes it out from the strand and makes sure that another one comes in, okay? So this is how DNA polymerases work. And the other thing is DNA polymerases will always work from 5' to 3'. Remember this, they always work from 5' to 3', which means if this is the new strand, this has to be working in this direction, right? If this was the new strand, then the DNA polymerase was working in this direction, which means if I have two strands, a 3' strand and a 5' strand, and its opposite strand, obviously I opened this DNA up, it's going to be like this. Now I'm going to throw in a few nucleotides and I'm going to ask you, which strand do you think DNA polymerase is working on? DNA polymerase always starts working from the 5' end. It can never start working from the 3' end. So if I throw in the DNA polymerases, they're going to attach, one will attach here, the other will attach over here and start working in these directions, right? So they will always work in the 5' to 3' direction. So if this is the DNA polymerase, where do you think this DNA polymerase is working on this strand or this strand? So it always works from 5' end to 3' end, which means this is the strand that was made new. This is the new strand that was being made. This is the nucleotide that was bringing in. These will be the bonds that were forming. Or alternatively, if the DNA polymerase was working from this end, then it would be possible. How? Because it's formed this phosphodiester bond, and when the active site came here, it realized these were mismatched, it excised the C and did not form a bond here. So very simple again. Not a complicated topic, just you just make a concept of it. So again, if I have two strands, a 3' strand and a 5' strand, and the opposite, a 5' strand and a 3' strand, the DNA, if I have only one, or if I say I have a DNA polymerase that has to, can only one of those that can work. So what's going to happen? So how is my DNA polymerase going to work? Now I'm going to change the direction of the arrows because I've been drawing the arrows 3' to 5', but now I want to draw them according to my DNA polymerase. So how am I going to make sure that you guys understand it? So what I'm going to do is every time I draw an arrowhead, I'm going to draw it according to the DNA polymerase, that is 5' to 3', and the opposite one is again going to be 5' to 3'. Now my DNA polymerase will attach over here and form these bonds in the direction of the arrow. What about this one? How is my DNA polymerase going to work here? I just told you that I've thrown in a DNA polymerase. This is the starting line, and I've thrown in a DNA polymerase here. How is my DNA polymerase going to work? Over here, it's very simple. It's going to work straight in a straight line and end up over here and having formed this second strand. This one shows my second strand, okay? How will I form a second strand on this strand? This is strand A, this is strand B. How will I form a second strand on strand B? If my DNA, for instance, I say there's one DNA polymerase, it's gonna work over here, then it's gonna have to work here, then over here, then over here. Okay, so how is it gonna be working alternating between these. What it basically does is the DNA polymerase, because obviously when it's working at this level, the rest of the strands are closed behind it. They have to open later, right? So it can't just go and skip over here and start working over here. No, it can't do that. It has to work in this area first, because we don't want all of the strands. Imagine all of the strands to the end are opened up, then there will be so much confusion. We don't want to do that. We want to open this small area, complete work here, then open this small area, complete work here, right? And keep stabilizing them. So how is my DNA gonna work over here? It can only attach from this end, which means it's gonna attach over to this end and complete work over here. Then when we reach this segment, segment 1 and segment 2, in this segment again, and this is segment 3, again, that's what's gonna happen. The DNA polymerase will attach over here and then complete work over here. Then in segment 3, again, it's gonna attach over here and complete its work over here. Now what ends up happening is that this strand, because it was a direct strand, what happened was it went directly and completed a whole layer that was totally complete. However, on this strand number B, which was the opposite strand for it, it became really difficult because there was one segment that was formed, then it had to move back, so this segment remained and this segment remained, which is why, because these occur in fragments. Look at these are fragments of the chain. They're not a complete chain, unlike this one. So what ends up happening is our DNA does complete it, but until the point that it isn't completed, scientifically, these are called the Okazaki fragments. Why are they called the Okazaki fragments? Because these are occurring in fragments of DNA. They aren't complete. So the man who discovered that this is what keeps on happening, that one window occurs, then another window occurs, then another window occurs, named these the Okazaki fragments. He was definitely called Okazaki, which is why he named them in such a way. So that is one thing you need to remember. The important things are function of the DNA polymerase, which is forming these bonds and excising incorrect bonds. The second thing is that a DNA polymerase always works from 5' to 3' and never the opposite. The third fact that you need to remember is prokaryotes and eukaryotes have different DNA polymerases. Prokaryotes have polymerase 1 and 3, okay? Whereas eukaryotes have differently names, one sigma and epsilon DNA polymerase, okay? So they have totally differently named DNA polymerases. They have alpha, gamma, sigma, epsilon, so differently named. And these prokaryotes, simple names, 1, 2, 3, eukaryotes, sigma, gamma, epsilon, etc. So again, we're gonna learn, I don't think we need to know details of this for A-level right now, but the important ones will come up either in the questions or in the book, and you can just learn which one is for which one. But right now, I want you to grasp the basic concept, which is 5' to 3'. If you remember 5', and the easiest way to remember is that 5 is the bigger number, so it's always gonna start from the bigger number, which is 5. Okay, again, we can see DNA polymerase is working here. Which direction is it working on? It's working in this direction, right? From 5' to 3'. So which means that this was probably the original strand. And okay, so which means that this was probably the original strand. And what it was doing is it's working to put in, excuse me, so what its DNA polymerase is doing is it's working to put in more of these nucleotides, right? It's all, and how does it work? It always works 5' to 3', okay? And so, okay, again, let's see. DNA is unzipped slowly over here. You can see it's being unzipped. The point where it is unzipped, and is becoming unzipped further is known as the replication fork, because it look at this, it looks like one of those tongs of the fork, right? So it's a replication fork. Again, as you can see, DNA polymerase can only work by 5 prime to 3 prime. Now what does it mean? It means that it's continuously working to arrange nucleotides in a 5 prime to 3 prime direction. And you know that when we have one new strand that is forming only from 5 prime to 3 prime direction, then the original strand it's forming against is obviously going to be the opposite or the anti-parallel of this one, which is 3 prime to 5 prime. So this is very important information. You need to be able to register this very well. What I'm saying is that the DNA polymerase attached over here can only connect nucleotides by forming bonds between them. These bonds can only occur if the 5 prime end of this nucleotide is here and 3 prime end is here. So if the new strand is forming 5 prime to 3 prime, which means the original strand should be 3 prime to 5 prime, right? Is that so? I hope that is clear. Again, even in the image we had previously, so look at this one. Now, what do we see? We see that the DNA polymerase is working in this direction from 5 prime to 3 prime to form this new strand, which means the original strand should be 3 prime to 5 prime, which is the exact opposite. So the direction of the DNA polymerase, you can appreciate over here, it's adding another one and another one. Okay, so this is basically what ends up happening. Okay, so again, because this is it's a continuous strand, because this is how it's happening, right? You know that I just told you that this area, this whole thing that's happening over here is called the replication fork, because replication is occurring and it looks like a fork. And the other thing that you also know is, now you need to know, okay, you also know that there's a continuous strand, which is obviously in the correct direction, because DNA's nucleotides can only be attached from 5 prime to 3 prime, right? And then you have a discontinuous one. So a discontinuous one essentially forms because we know that we can only attach the DNA polymerase from a 5 prime to 3 prime end. However, over here you can see that if this is the 3 prime end, 5 prime end, then the 3 prime end, right? So this means that as the DNA polymerase attached here, it formed a continuous segment here. But when it came here, it realized that, okay, this strand runs 5 prime to 3 prime, right? This strand runs 5 prime to 3 prime, which means I can't, and if I can only attach 5 prime, then I have to move over here, attach over here during this window, and form a 5 prime nucleotide over here, a 3 prime again to make sure that the 3 prime is here. Then in the next window, I have to again move back over here. Sorry, this is how it's okay. This is the 3 prime and this is the 5 prime. Yeah, okay. And then I have to move back here, and then when I'm here, I have to move back here to make sure that what I get is over an overall direction opposite to this one, right? Overall direction of this strand, of the discontinuous strand, should be 5 prime to 3 prime, because this one is 5 prime to 3 prime in this direction. So what ends up happening is discontinuous strands are formed, which are known as the Okazaki fragments. Okay, so these are the discontinuous strands that form. So now we have this diagram, which we essentially need to understand every single enzyme on this one. Not probably every single enzyme, but the important ones that we're going to talk about just now.